The Venom Lab experiment. We did an experiment with rattlesnake venom, testing to see the effect of rattlesnake venom in different temperatures, such as room temperature, ice, and boiling water. The search methods. During this project, we learned about different search methods to use in a crime scene, the effectiveness of different methods, and the types of crime scenes the search methods are used for. The crime scene. This project, we were introduced to Anna Garcia. We collected evidence at the crime scene. We took pictures, measurements from the point of origin to certain items.
It is very important to try to find patient zero. Patient zero is the first person to contract whatever disease is spreading. Finding this person may make it easier on how the person got such disease and help find a cure much faster. During our weightlifting experiment, we were trying to see how our energy levels dropped throughout the workout. We concluded that by the last set of our exercise, we had to use as much effort as possible to complete the workout properly. Drawing blood is beneficial to determine if someone has any diseases. You can test someone's blood in a sterile lab. From the results from these tests, you can help prevent any serious condition you may have, as well as seek any treatments if you have any such diseases.
Testing anastomach contents. We did this experiment to test whether anastomach contents contain polysaccharides, monosaccharides, or polypeptides. We also did this to observe a change in the chemicals when they are exposed to vomit. We used chemicals such as iodine, Benedict's, and Bleuret. We used a polysaccharide such as starch, a monosaccharide such as corn syrup, and a polypeptide like protein powder. Iodine and starch gave us brown to black, Benedict with the corn syrup gave us blue to orange, and the Bleuret with the protein powder gave us blue to black. This was very valuable because it helped us to get more experience with the chemicals we were using. It also helped us to learn the difference between a monosaccharide, a polysaccharide, and a polypeptide. This was also useful because it helped us to learn whether anastomach contents contain any of the chemicals we used or not. Calorimetry. We did this experiment to see how many calories were inside of a nutrition label. We took a can, we filled it halfway with water, then we put a thermometer through the opening of the can. We then placed our heated food under the can. This was to see how much the water changed in temperature. This experiment was very valuable because it taught us the difference between a kilocalorie, a food calorie, and a chemistry calorie. This also helped us to see how much food we were actually consuming versus what it actually says on the nutrition label. We used math equations and formulas. This was to show us the result of the total calories we were consuming of the nutrition label we were working on in class.
During the school year, we did a lab called gram staining. Gram staining is where we better understood the difference between gram-positive and gram-negative and what colors they are based on the chemicals we used. During the school year, we did a lab called the blood pressure lab. The blood pressure lab is where you take someone's resting heart rate and then you do something that you believe will increase their heart rate, and you take their heart rate again afterwards to see if the heart rate has increased or decreased. We did this to see the difference in resting heart rate and active heart rate.